where does transcription occur? In the nucleus, right? In prokaryotes, where does transcription occur? In the cytoplasm. Good. You guys are doing great. Okay, and now we're ready to discuss the last step in structural gene expression. The step we call translation. And you guys, the translation is like an onion. There's so many layers to it. And what we're going to do, just to get ready for the exam, we're just going to do the first superficial layer. We're going to make sure you can translate an mRNA sequence into a protein, right? And then all the details of this incredible process we'll be describing after exam 2. So the details will be on lecture exam 3. We're just going to do the superficial part today. So folks, where does translation occur in prokaryotes and bacteria? In the cytoplasm. Where does translation occur in eukaryotes like your cells? Okay, maybe I said that wrong. Okay, she might have said that wrong. Okay, where does translation occur in eukaryotic cells like your cells? Where does Yeah, the ribosomes, and the ribosomes are where? In the nucleus, or are they in the cytoplasm? In the cytoplasm, right? And I'm aware, you guys, that you have rough ER with ribosomes on it, but initial binding of our ADS ribosomes to our messenger RNA occurs in the cytoplasm. So we will say that translation occurs in the cytoplasm of both prokaryotes and eukaryotes. Okay, good, you guys. All right. So now we're going to do that final incredible process. We're going to take the information encoded in the base sequence of our mRNA, and we're going to translate it into the amino acid sequence of a protein. An incredible process. We want to remember that the sites within the cells where translation occurs are the ribosomes. Good. Okay. And again, we're going to do this very superficially. So the key to translation is the genetic code. And this is where the second pink handout, you guys, from last time. This is where we're going to discuss the genetic code. And then we're going to practice using the genetic code by translating a short sequence of messenger RNA that's on the back of your handout. Okay, so first let us explore this incredible genetic code. So a definition of the genetic code is the one-to-one relationship between a codon, and codons are 3 bases of mRNA. The genetic code is a one-to-one relationship between a specific codon and a specific amino acid. And there'll be a few exceptions, but we'll just use that as our general discussion. Okay, so here's some key terms. So the mRNA is going to be translated in codons. And again, codons consist of 3 bases of mRNA. So let me just, let's use an example. Yeah. We're still staying prokaryotic. Okay. The genetic code is universal, however, so most cellular organisms use the same genetic code with some variations. And the fact that all cellular organisms use basically the same genetic code tells us it evolved so early in the evolution of life. Yeah, so this is often described as a universal genetic code from the lowest bacterium to a human. We're using basically the same genetic code, which is just incredible to think about. Yeah. Okay, but again, folks, we're gonna stay focused on prokaryotes, on bacteria. Okay. So again, bacterial translation. And a simple definition of bacterial translation is mRNA acts as a template to make what? Proteins. Good. Remember, we're switching languages from nucleic acid language to language of proteins. Okay, so this is translation. And we're just going to dive into our example here. You guys sometimes ask these. So we said that the mRNA will be translated codon by codon into amino acids. So what the heck are we talking about? So this is our mRNA, 5 prime to 3 prime. And I'm just going to add just a couple of details, you guys. So on our messenger RNA, there will be a special base sequence called the RBS. And RBS stands for ribosomal binding site. Okay, so its function is similar to the promoter in transcription. Okay, our ribosomes must start at the beginning of the mRNA for a protein, right? We don't want our ribosomes to start in the middle of the mRNA encoding the protein, because we just end up with like half a protein. So we have to have special mRNA sequences that our ribosomes will recognize. And again, I'll speak with you here. So the ribosomes are scanning mRNA until they hit the ribosomal binding site. They're going to bind, and then they start moving downstream until they hit what we call a start codon. So let's just put our start codon in. The most common start codon is AUG. So here, folks, I use the term codon. Okay, a codon is 3 bases on mRNA, and each base has a position. So this is the base in position 1. This is the base in position 2. This is the base in position 3. Now, the beauty of the genetic code table is it tells me which amino acid will the ribosome translate this mRNA codon into. So what we want to do is we want to take a look at our genetic code table, and this will be on the exam, you guys. This or a similar genetic code table will be on the exam. But you must know how to use it, right? I'm going to ask you to translate an mRNA into an amino acid sequence. This will be on the exam, but you have to know how to use it. So let's just get oriented. Here we see all the different codons possible in mRNA. And we see in each box, it's telling us which amino acid a specific codon is translated into. Now, in this genetic code table, they're only using the 3-letter abbreviations for amino acids. Okay? On the genetic code table I give you on the exam, it will have both the abbreviation of the amino acid and the full name. On the exam, it's permissible just to use the 3-letter abbreviation, okay, just so you know. All right. Now, the way the genetic code table is organized, along the left-hand column, this is the base in the first position. Along the top is the base in the second position. And along the right-hand side is the base in the third position. Okay, so let's just take a moment, you guys, and see if we can figure out this codon AUG. The first AUG the ribosome encounters is described as the start codon. This is where translation actually begins. Okay, so A is in the first position, U in the second, G is in the third. So let's see what we can find here. So first position is A. So I know my codon is in this row. The base in the second position is U. So I know that my codon is going to be at the intersect of A and U. And usually it's just easiest by inspection. There's my codon AUG. So AUG is translated into the amino acid, MET stands for methionine. Okay, so I'm just going to go ahead and I'm going to make a little polypeptide chain here. Clean this up a bit. So the ribosome is going to translate AUG into which amino acid?
MET.
MET. MET stands for methionine. Now here's a little tidbit. We said the first AUG encountered by the ribosome, we call that the start codon. In prokaryotes, the first AUG is translated into a special chemically modified methionine called formal methionine. Now, on the exam, you don't have to remember that, so this would be only in prokaryotes. The start codon AUG is translated into a chemically modified methionine called formal methionine, or F-MET. So I'm just going to put F-MET here. On the lecture exam, This will be another short answer question. I'm going to give you a DNA sequence. You're going to transcribe it in mRNA sequence, and then you're going to translate it into amino acid sequence using your genetic code table. On the lecture exam, you guys, it will be in bacteria. If you don't put FMET, I'm not going to take any points off. Okay, you can just put methionine. In eukaryotes, like our cells, the first AUG, the start codon, is translated simply into methionine. Okay, so it's a little tidbit, and I don't want you to get stressed about it too much. Okay. Any subsequent AUG codon is simply translated into methionine in bacteria. It's only the first one that gets translated into FMET. Okay. So we're going to practice now, you guys, and we're going to practice using the example on your handout. The little circle there that's translating the mRNA, what's that little circle represent? The ribosome. And you can see we're doing this so simplified, right? And that's the key for right now. Okay. So you guys, on our mRNA, what's the next codon? Okay, UCU. UCU. So that's our next codon, right? UCU, first, second, and third. So take just like 5 or 10 seconds and see if you can use your genetic code table to find the amino acid that the mRNA codon UCU will be translated into. So did you guys come up with SER? Mm-hmm. Okay, that's serine, the amino acid serine. So the next amino acid would be SER. This stands for serine. Okay, good. What's the next codon on the mRNA? It's hard to read, isn't it? That's G. Okay, so this is a codon. And now the ribosome will move down to the next codon, G, U. Okay. So folks, again, practice using your genetic code table. If you're having trouble, would you just turn to your neighbor on either side and see if they might be able to help you? So you guys, how am I going to find a G, first position? G, what's the next one? G-U. G-U-U. So that would be translated into, val means valine, amino acid valine. So the next amino acid would be val or valine. Good, you guys. All right. Now I'm going to change the our sample mRNA here, and I'm going to skip the next 2 codons, and I'm going to introduce a codon. Okay, so I'm changing this a little bit, you guys. So let's pretend this is the next codon. UAA. So help me out here. Which amino acid will UAA? Here's U, there's A, there's UAA. So which amino acid does UAA encode? Nothing, right? This is called a stop codon. A stop codon, and you can see there's 3 stop codons in mRNA, UAA, UAG, and UGA. What do you think happens? And again, we're simplifying this, you guys. What do you think happens as the ribosome is moving along the mRNA, translating each codon into a specific amino acid? What happens when the ribosome hits UAA? It terminates. It stops translation. And again, folks, we're just going to simplify. We're going to say when the ribosome hits a stop codon, it releases the mRNA, it falls off, and it releases the polypeptide. Okay? So this is our stop codon. So ribosome falls off mRNA, thus stopping translation. And not only does the ribosome release the messenger RNA, it also will release the polypeptide that it's just finished synthesizing. You guys, is it making sense? Okay, so let me give you an example of what you're going to see on the short answer section of exam 2 to make sure we can do this. Okay, so on the short answer section, I'm going to give you a DNA sequence, And you guys, I'm not trying to trick you. I'm gonna set up the DNA sequence correctly. So some people tell me later, oh, I thought you were trying to trick us and you set the DNA sequence up backwards. I go, no, I don't need to trick you. It's hard enough as it is. So I'm just gonna make up this DNA sequence. Okay, so we're gonna say this is E. coli. So let me see if I can do this right. So this would be our DNA sequence. And I'm just going to randomly do this. Okay, you guys. So I'm going to give you a DNA sequence. And again, I'm going to set it up correctly. I'm not trying to trick you. And I'll say transcribe the DNA sequence. And we're presuming this is DNA, the DNA sequence of a structural gene, a gene that encodes information for a protein. Okay, so when I say transcribe the DNA, what's going to be your product? Good, mRNA. Good, you guys, excellent. Okay, so what would be the base sequence of your mRNA? And remember, you guys, I'll set up the DNA correctly so that your mRNA will. You just do transcription, and it's all set up correctly. I'm not trying to fool you. Okay. So what's going to be the base sequence of my mRNA when you transcribe your DNA? What are we going to have? Good. Okay. Good. 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 But you can't see. So it's 2 A's. Oh, what am I doing? Wait a minute. Oh, I messed that up. I didn't want that. Let me see here. How can I do this? Oh, I screwed this up, you guys. I'm so sorry. I'm sorry. Okay, now let's see if I did that right. Okay, so when you transcribe this ATT DNA into okay, all right, you guys. So you want the question then will say, now translate the transcription product. Okay, so what does translate mean? I'm going to translate my mRNA codons, and I'll just put little brackets around the codons, into what? What's my, if I have mRNA and I translate it, what's going to be my product? What would we call it? The protein or polypeptide? Good. Okay. So you guys, let's help me out here. AUG. Oh, come on. This is so exciting. Okay. So again, if you put, synthesis is equal, like if you put FMET for the start codon, maybe I'll give you a bonus point, but you don't have to, right? You don't have to. Okay, so FMET is our first amino acid, right? What's our second amino acid? AAG. AAG. LYS is lysine. S is lysine. What's the next amino acid, you guys, in your translation product? LU. CUG. CUG is LU is leucine. What's the next amino acid? Stop. Good, you guys. UAA. UAA. Stop. All right. So you can just finish it there, or you can just simply put stop, or you can just put nothing. So, folks, that will definitely be a short answer question on the exam. Make sure you can do it. If you're having trouble with it, touch base with me. Yeah.
What does the F stand for?
The F is in bacteria in prokaryotes. In prokaryotes, the first AUG, the codon, the start codon, is translated into formal methionine, a chemically modified methionine. Eukaryotes, start codon is just translated into methionine. So we said you don't have to put the F. Maybe we give you half a bonus point or something if you did. Okay, but you don't have to, because I know you guys are just so overwhelmed. You guys okay with this? Yeah.
Yep, they have to put the full name or just under.
On the exam, you can use the 3-letter abbreviation. The genetic code table, you guys, it's on the exam. It has both the 3-letter abbreviation and the full name of the amino acid, but you can use the 3-letter abbreviations. That's fine. Yeah. You guys okay with this? All right. And then, as I said, on the study guide, there's all kinds of additional questions on translation. We'll hit those topics after lecture exam 2. And the finer details of translation, you will see questions on lecture exam 3 about it, but you won't see those details we haven't discussed on lecture exam 2. Oh, there's one more thing though. I keep saying that, right? Oh, one more thing. Oh, there's one more thing. Oh, there's one more thing. There is one more thing. So, do you remember, folks, prokaryotes, bacteria, what kind of ribosomes do bacteria have? 70S, right? And eukaryotes, what kind of cytoplasmic ribosomes do we have? 80S, right? Why do we care? Well, it turns out that 70S ribosome is a wonderful target for antibiotics. And I have to put cytoplasmic ribosome here. So in our cytoplasm, we have the big 80S ribosomes. The reason this is important is that many classes of antibiotics specifically target, inhibit the bacterial 70S ribosome. So I would like you guys to remember from chapter 4, those classes of the tetracyclines. The class called macrolides, which includes erythromycin. Aminoglycosides, and an antibiotic that doesn't get used much because it can cause bone marrow suppression, but in emergency cases they might use chlorantraniliprole. These are four classes of antibiotics that specifically inhibit what? Yeah, these inhibit the 70S ribosome. So which process would not occur? Protein synthesis or translation. Good. Why is it, you guys, if we're taking these antibiotics, why isn't protein synthesis shut down in our cells? Yeah, our ribosomes are different enough that these antibiotics at therapeutic levels, safe levels, are going to specifically target the smaller 70S bacterial ribosome and will not cause inhibition of protein synthesis or translation on our larger 80S ribosomes. So that's kind of important. Okay, folks. Yeah. Okay. We'll do more details of this after exam 2. So just that what we just did, you guys. That's the end of information for exam 2. And what I thought I would do here is quick quiz. Yeah, I'm sorry. Yeah, tell me.
So the slides that we didn't cover in between all these, are we responsible for this as well?
If there was information that I verbalized, you're responsible. If there's information on the slides that I didn't verbalize, you aren't. Okay? Okay, you guys. I know this is like you're doing backflips in your head, but quick quiz. Okay, what is the function of bacterial gyrase? It relieves supercoiling, right? Okay, which group of antibiotics will inhibit bacterial gyrase? You're there. You guys are there. You got it. So the class, the big class is called quinolones. There's a subgroup called fluoroquinolones. And the specific one we talked about was what? Ciprofloxacin, right? So if you have a patient, say, that's infected with Bacillus anthracis, a bacterium that causes anthrax, and you give them ciprofloxacin, why will that inhibit the growth of the Bacillus anthracis? Good. If the bacteria can't relieve supercoiling, can they carry out chromosomal DNA replication? No. Can they transcribe their chromosome? No, right? Okay, good. Excellent, you guys. What's the function of helicase?
Break hydrogen bonds.
Good, good. It unzips a double-stranded DNA. It breaks the hydrogen bonds between double-stranded DNA to create what? Single-strand templates. Excellent. What are SSBPs? Single-strand binding proteins. What is their function? Right, they're going to protect the single-stranded DNA from reannealing, from tangling, and from hydrolysis by bacterial enzymes that will destroy single-strand DNA. Good, excellent. What is primase? What is it? Yes, it's a special RNA polymerase. What's its function in DNA synthesis? Excellent, good. It's going to synthesize that short little RNA primer that then permits which enzyme to start work? DNA polymerase 3, good. So DNA polymerase 3, what is it making? Yeah, DNA. And it's synthesizing in which direction? I mean, the new strand is being synthesized in a 5' to 3' direction, right? What's orientation of the new DNA strand to the template? Anti-parallel. Dynamite, you guys. Okay. What can DNA polymerase 1 do that DNA polymerase 3 can't do? Good. DNA polymerase 1 can remove the RNA primer and replace it with DNA. Good. On the leading strand, the leading strand of DNA is made using which kind of DNA synthesis? Continuous. Okay. The lagging strand is made using discontinuous. Good, you guys. What on the lagging strand? What do we call the little DNA fragments that are a result? Okazaki fragments, right? And which enzyme is going to covalently link the Okazaki fragments together? Yeah, good. DNA ligase. You just nailed it. You just got 100%. That is so cool. Okay, so just other terms for every guys. So the leading strand, continuous DNA synthesis, lagging strand, discontinuous. Okay, right. Oh yeah. Okay, good stuff here. Yeah. Okay. Yes. You guys are just going to do so good. I can feel it. Okay. So those of you I want to see in the lab, have a good weekend. Try to eat. Try to sleep the night before the exam. Okay. As always, you guys, it will be a long exam. So remember your job. Just get through that exam once, and then go back and talk about the questions. Can't feel it.
Let's not do those on my way out.
Yeah, you